Okay, so today we will learn the chapter 16, regulation of gene expression. Chapter 16 is composed of the 5 subjects starting from the prokaryotic gene expression is regulated in operon. Finally, we will cover eukaryotic gene expression can be regulated after transcription. So in this slide, we will learn the first subject. Prokaryotic gene expression is regulated in operons. First of all, we have to learn what is negative regulation and what is positive regulation. Okay, so this is the example of negative regulation. Okay, so DNA has the repressor binding site. Okay, so if there is no repressed proteins, transcription happens. Okay, however, in this case, repressor proteins bind to repressor binding site. Okay, it means that binding of repressor proteins blocks transcription. Okay, this binding blocks transcription. So this is, we call it as negative regulation. Okay, how about the positive regulation? So this is the DNA. So they also have another binding site. So this is the activator binding site. So without activator proteins, there is no transcription. However, binding of activator proteins stimulate the transcription is like this. So this is, we call it as positive regulation. Okay, so a little bit different from the negative regulation, positive regulation. Okay, so this binding blocks transcription. However, this binding stimulates transcription. Okay, so in this slide, we will learn what is inducer. Okay, so inducer is compounds that stimulate protein synthesis. The very good example is lactose. Okay, let me see what is lactose. So lactose is disaccharides of galactose and glucose. Okay, they have 2 saccharides. Okay, and what is beta-galactosidase? So the main function of beta-galactosidase is cleaves lactose to form galactose and glucose. And finally is the glucose. So glucose is the easiest sugar to metabolize. So usually the glucose is directly used in the glycolysis. Okay, so I mentioned that lactose is inducer. Okay, for one case there is no lactose. If there is no lactose, beta-galactosidase is not synthesized. However, if there is a high lactose, they have lots of lactose, it means that it can induce the beta-galactosidase is synthesized. Okay, so we call it as a lactose as an inducer. Okay, let me go to the inducible proteins. So inducible protein is that protein synthesized by inducer stimulation. Previously we mentioned that what is inducer? Lactose was inducer and beta-galactosidase is synthesized or is not synthesized. So beta-galactosidase is the inducible proteins. Okay, let me see here. So x-axis is time and the y-axis is level of beta-galactosidase. After we add inducer, after we add lactose, okay, so the level of after we add inducer, the level of the beta-galactosidase is increased is like this. Okay, so after we remove the inducer, so this is the beta-galactosidase level is the maintained is like this. Okay, the important thing is that lactose inducer induces the synthesis of beta-galactosidase. Okay, so it means that beta-galactosidase is inducible proteins. Okay, so in this slide we will learn the regulation of metabolic pathways. Previously we learned that what is metabolic pathway. So metabolic pathway is, we call it as the post reaction is commitment step. After that, other reaction that happens in sequence. So at that time we learned feedback inhibition. It means that final product act as a non-competitive inhibitor of post enzyme. Okay, so allostatic regulation means that so this is precursor. They need enzyme 1, enzyme 2, enzyme 3, enzyme 4, enzyme 5, and they will make the final end product. So this end product act as non-competitive inhibitor of post enzyme. Okay, after that they shut down the pathway. So this is the allostatic regulation. Okay, another regulation is recorded as transcriptional regulation. For example, in this case, okay, this is end product. They need enzyme 1, enzyme 2, enzyme 3, enzyme 4, enzyme 5. So enzyme 1 is encoded by gene 1, enzyme 2 is encoded by gene 2, enzyme 3 is encoded by gene 3, and enzyme 5 is encoded by gene 5. So transcriptional regulation means that end products blocks the transcription of intermediate gene is like this. So this is the we call it as transcriptional regulation. A little bit different between the allostatic regulation and transcriptional regulation. Okay, so allostatic regulation is end product act as a non-competitive inhibitor of only first enzyme. However, transcriptional regulation is blocks the transcription of the intermediate gene is like this. Okay, so this is the transcriptional regulation. Okay, so in this slide we will learn what is operon, and also we will learn how prokaryotic gene expression is regulated. Okay, first of all we have to learn what is operon. So operon is a gene cluster with a single promoter. Okay, mainly operon is composed of the promoter region and also operator region, and they have the several structural genes. It's like this. Okay, but main thing is one operon has one single. They only have one single promoter. Okay, so what is promoter and operator? So promoter and operator is placed where regulatory proteins binds. Okay, and what is a structural gene? For example, in this case, in this RAC operon, structural gene is beta-galactosidase and also galactoside polymerase. Finally, there is a galactoside transacetylase. Okay, so we already know that what is galactosidase. The main function is the cleaves the lactose to make the galactose and glucose. What is main function of polymerase? Okay, so this is cell membrane. Okay, to transport extracellular lactose to inside the cells, we need channel, we need a hole. So this is the polymerase, is a channel protein. Okay, so they help the transport of extracellular lactose into the cell. And what is the galactoside transacetylase? Okay, so usually the beta galactose, so beta galactose can be changed to the beta galactoside. Okay, but however, beta galactoside is a little bit toxic. So the main function of this one is a detoxify beta galactoside by transferring acetyl group. They add acetyl group here, so it means that it can detoxify beta galactoside. Okay, anyway, they have a promoter and operator. They have a structural gene is like this. Okay, so this is the operon. Okay, and also lock operon is inducible system. Okay, when lactose is absent, they have some function. And next slide we will learn when lactose is present. Okay, let me start from when lactose is absent. Okay, so this is the lock operon, promoter and operator. They have the structural gene is like this. Okay, so in front of the promoter, they have gene I. So gene I is the main function is encode repress proteins. Okay, except that this gene is transcribed. After that, they will make the active repressor. So this active repressor bind to the operator region. Okay, so the bound repress proteins prevent RNA polymerase from binding to the promoter region. So in this case, transcription is blocked. So previously we learned that negative regulation. Okay, so binding of repressed proteins to the repressed binding site, it blocks transcription. Okay, in this case, is also active repressor bind to the operator region. Okay, it blocks transcription. Okay, so it means that when there is no lactose, repressed protein bind to the operator, it blocks transcription. So, okay, so in this slide we will learn what happens when lactose is present. Okay, so this is the allolactose function as inducer. So what is allolactose? Okay, so this is lactose. Lactose can be changed to the galactose and glucose. But sometimes the lactose is changed to the allolactose. So allolactose can be cleaved to the galactose and glucose. Okay, so in this case, allolactose bind to the repressor. So converting the repressor into the inactive repressor. So in this case, inactive repressor cannot bind to the operator region. So that's why RNA polymerase can bind to the promoter region. After that, it can start transcription is like this. It can start transcription is like this. Okay, so when lactose is present, lactose can be easily converted to the allolactose. So usually the allolactose function as inducer. It binds to the repressor. It will make the inactive repressor. So it cannot bind to the operator. Usually at that, usually the RNA polymerase can bind to the operator region. It can start transcription. Okay, so in this slide, we will learn the new concept, the amount of catabolite regulate the rock operon. Okay, so what is catabolite? Okay, for example, in this case, lactose is changed to galactose and glucose. So we call it as galactose, and glucose is catabolite. Okay, so in this case, we are thinking about the amount of catabolite, especially the amount of glucose. Okay, so when glucose concentration is very low and lactose is present, so in this case, CAMP is produced. Okay, so at that CAMP bind to the CAMP receptor proteins, we call it as CRP. At that CRP and CAMP complex bind to the polymerase region. Okay, so this binding is it induced efficient binding of RNA polymerase. At that, it will increase transcription. Okay, so this is one case when the amount of glucose is very low. Okay, so in this slide, we will learn when the amount of catabolite is high. Okay, so in this case is high glucose concentration. The catabolite, especially the glucose amount is high and the lactose is present. In this case, CAMP is not produced. So in this case, because CAMP is not produced, so CAMP cannot bind to the CRP. Okay, so in this case, CRP will become inactive status. So in the inactive CRP cannot bind to the polymerase region. So without CRP bindings, RNA polymerase also cannot bind to the polymerase region. So it means that there is no transcription. Okay, so previously we learned that when the amount of catabolite is very low, so in this slide we learned that when the amount of glucose is high, so there is no transcription. Okay. Okay, so in this slide we will learn the second subject, eukaryotic gene expression is regulated by transcription factor. So this picture shows that potential point for regulation of gene expression. So we can regulate gene expression before transcription and also during the transcription and also after transcription but before translation 3, 4, 5. Also we can regulate gene expression at translation and also after translation. So we will see some details in the next slide. So in this slide we will learn the transcriptional factor can regulate transcription. Okay, so this is we call it as transcriptional control. Okay, let me see some details. Okay, so usually this is DNA. DNA is composed of promoter and coding region. So usually the transcription factor 2D is bind to the Tata box region. Okay, so after that it bind to the Tata box. Next, transcriptional factor 2B, it bind to the here. So it bind to the here. So this bindings help to recruit RNA polymerase 2. After that, RNA polymerase 2 is bind. After that, transcriptional factor 2F, 2E, 2H is bind to the here. So they made basal transcription apparatus. Okay, so they made the basal transcription apparatus. Okay, next, two activator is involved in the transcription. Okay, first of all, so one activator is located bind to the here. Another activator is bind to the enhancer region. Okay, so one activator is bind to the promoter region. Okay, so the other activator is bind to the enhancer region. Okay, after that, the mediator bind to the basal transcription apparatus. After that, there's DNA is bent, so linear DNA is bent. So DNA bending allows distantly bound transcriptional factor to interact with the mediator. Okay, so for example, this activator is they are far away from the transcriptional apparatus, basal transcriptional apparatus. Okay, however, the DNA bendings allows distantly bound transcriptional factor to interact with the mediator. After that, it can initiate transcription. So it can initiate transcription is like this. Okay, so let me summarize it. So transcriptional factor. So there are several kinds of transcriptional factor, starting from transcriptional factor 2D, B, F, E, H. After that, two activator and mediator, DNA bending. So this is the can regulate transcription. So it can initiate transcription. Okay, so in this slide we will learn another concept. So environmental signal can regulate transcription. For example, environmental signal, for example, drought. So it means that there is no water. Okay, for example, stressor, so some kind of drought can activate transcription of gene that encodes a transcription factor. So this stressor can activate transcription of gene that encodes transcription factor. Okay, so usually this transcriptional factor can bind to the SRE. SRE is a stress response element, which is located in the promoter region. Okay, so this is located in the promoter region of gene A, and also this is located in the promoter region of gene B, and also this is located in promoter region of gene C. Okay, so this transcriptional factor bind to the SRE. Okay, so this binding can stimulate transcription of gene A and gene C, gene B and gene C. Okay, so it can stimulate the transcription of gene A and gene B and gene C. Okay, so gene A made protein A and protein B and protein C. So each protein is participate in this drought stress response. Okay, so if there is no water, maybe a plant should will be the length will be increased because there is no water. Maybe so this protein A, this protein B, protein C is involved in this processor, I think. Okay, so let me summarize again. So environmental signal can regulate transcription. For example, one case is drought, so it can activate transcription of several transcription factors. So it binds to the SRE elements, so it can stimulate transcription of gene A, gene B, and gene C. And this protein is participate in stress response. Okay, so in this slide, I will explain the third subject. Virus can regulate their gene expression during reproductive cycle. So what is virus? Okay, so this is virus structure. Okay, they have the viron, so they have the DNA or they have RNA. Okay, after that they have protein coat is like this. The virus, we call it as infectious microbe consisting of viron. So sometimes virus has DNA or sometimes virus has RNA, and also they have the protein coat is like this. And also, what is bacteriophagy? So bacteriophagy is the structure is like this, their shape is like this. Okay, we call it as bacteriophagy is a type of virus that impacts bacteria. Okay, so this is bacteria. So bacteriophagy can only infect the bacteria. Okay, so this is their shape, like this. Okay. So in this slide, we will learn how virus regulate their gene expression during their lytic life cycle. So we will see in this slide and next slide what is lytic cycle. Okay, so let me start from this bacteriophagy can impact the bacteria. So virus can impact host cells. Okay, after that they inject their viral DNA. Okay, so virus, they don't have RNA polymerase, so they cannot start their transcription, but virus uses bacterium's RNA polymerase to transcribe early gene in the viral genome. Okay, so this is viral genome. They are composed of the early gene, they are composed of the late gene. Okay, so in this case, they used bacteria's RNA polymerase to transcribe the early gene. So early gene is composed of, they have the several protein, it's like this. They have protein A, protein B. They can translate protein A, protein B, and protein C is like this. Okay, so one early protein can shut down bacterium itself transcription. Okay, so bacteria itself, they can initiate their own transcription, but one early protein shut down the bacterium's transcription. Okay, after that another early proteins, another early proteins stimulate viral genome replication. Okay, previous proteins, they shut down bacterium's transcription. Okay, another early proteins stimulate viral genome replication, and also another early protein can stimulate the transcription of late gene. Okay, so for example, in the late gene, they can translate it to the Purge protein and also capsid, or while the viral enzyme is like this. So which lead to production of new viral capsid protein and also Purge protein, and the Purge enzyme is like this. The important thing is that another early protein can stimulate the transcription of late gene is like this. After that, bacteria is burst Okay, so initially the one bacteriophage impacted. Okay, after that, there are several kinds of, there are lots of bacteriophage is synthesized. Okay, so this is recorded as a lytic cycle. So finally, they will burst bacteria. Okay, after that, hundreds of bacteriophage can be synthesized is like this. So this process we call it as a lytic cycle. Okay. So let me go to the how virus regulate gene expression during lysogenic cycle. So what is a lysogenic cycle? Okay, sometimes bacteriophage impact host bacteria. Okay, so they inject their phage DNA. Okay, but sometimes phage DNA incorporated into bacterial genome. Okay, so it becomes non-infective prophage. So in this case, they become the non-infective prophage. Okay, so this one will become prophage. So this is the non-infective. So at that bacterial chromosome with prophage can be replicated. Okay, so 1 will become 2 and 2 will become 4. 4 will become 8 is like this. And at that, under certain condition, prophage itself exercise from the bacterial chromosome. At that, it starts a lytic cycle is like this. Okay, but however, this procedure we call it as the lysogenic cycle. Okay, they are non-infective status. Okay, so they are incorporated into the bacterial chromosome. Okay, so usually the bacterial chromosome with the prophage is replicated. Okay, 1 become 2, 2 become 8. Okay, and after that, under certain condition, prophage itself exercise from the bacterial chromosome. It will start lytic cycle is like this. Okay, so this is the lysogenic cycle. So this is the how they regulate gene expression. Okay. Okay, so in this slide we will learn the fourth subject, epigenetic changes regulate gene expression. Okay, so this concept is belong to the remodeling of chromatin. Okay, so usually the DNA methylation. In the next slide we will see the histone acetylation plays very important role in the remodeling of chromatin. Okay, let me start from the DNA methylation. So what is DNA methylation? So DNA methylation can regulate transcription without changing DNA sequence. Okay, only thing is that DNA methylation occurs in cytosine. Okay, so this is cytosine. So methyl group can be added into here. So it will make the 5-methyl cytosine. But cytosine is changed to 5-methyl cytosine. Still they are cytosine. So there is no changes in DNA sequence. The only thing is methyl group is added. Okay, so for example, usually the methylation happens in the cytosine. Okay, so when there is a CG, so in this case, especially in this C, is easily methylated. Okay, so we call it as this CG. We call it this CG as, especially we call it as a CPG. Okay, this CG, okay, usually the cytosine is the methylated, so when they are located like the CG like this, okay. So usually the DNA methylation can be replicated, so they will make the one is also can be methylated and the other also can be methylated. Okay, so DNA methylation can be replicated is like this. So I will not teach you the details information, but important thing is DNA methylation can be replicated. So it means that DNA methylation can be inherited to your offsprings. That's why we call it as genetics, epigenetics. Okay, so what happens when cytosine is methylated? Okay, for example, usually the promoter region, they have lots of CG repeat. Okay, so usually when methylation in cytosine is happen in promoter region, so if there is no methylation, usually promoter RNA polymerase can bind to the promoter, it can activate transcription. However, methylation happens in promoter region, it prevents RNA polymerase from binding to the promoter region, so there is no transcription. Okay? So in this slide, we will learn how DNA methylation changes development of bees. Okay, so this is our chapter 16. We can see lots of bee, but this is the king bee and this is the work bee. But important thing is king bee and work bee, they are starting from same larvae, it's like this. Okay, so one group we inject, in control group we inject the solvent, so the other group we injected DNMT inhibitor. So what is DNMT? So DNMT is the full name is DNA methyltransferase. So this is cytosine. So DNMT add methyl group, so it will make methylate the cytosine. However, if we injected the DNMT inhibitor, so there is no methylation. Okay, so if there is a methylation it happens, so this larvae will become the work bee. However, if there is no methylation, the same larvae will become the queen bee, is like this. So usually the royal jelly is function as the royal jelly has the DNMT inhibitor. So royal jelly can inactivate DNMT, so it means that there is no methylation, so it means that it will become the king bee. So if we did not give the royal jelly, it means that it will become a work bee, is like this. Okay, so it means that in this slide we will see that DNA methylation plays important role in development of the bee. Okay, so it can become the work bee or it can become the king bee. Okay. Okay, so in this slide we will learn another epigenetic changes, histone acetylation. So this is the histone and this is the DNA, is like this. Okay, histone has lots of amino acid. Okay, histone acetylation means that add acetyl group into amine in lysine. So this is the amino acid lysine. Okay, and finally they have NH3. This is, we call it as amine. Okay, usually the amine is the positively charged, and also DNA is negatively charged. So it means that histone proteins and DNA is closely interacted. However, so if we add acetyl group, so this is the acetyl group. Okay, this is acetyl group is added. Okay, it means that there is no positive charges. Okay, so there is no positive charges. It means that result in weakened interaction between the histone and DNA. Okay, previously, when there is positive charges, they are closely interacted. Now they will become the weakened interaction between the histone and DNA. They show that relaxed chromatin. So when the chromatin exhibit very relaxed structure, so it means that transcription is actively happens. So it will enable active transcription is like this. Okay, it means that epigenetic changes, especially the histone acetylation, activate transcription. So in this slide, we will learn the heterochromatin and euchromatin. So previously we learned histone acetylation, and histone acetylation make the relaxed chromatin. However, without histone acetylation, chromatin is very compact. They showed very compact structure. Okay, heterochromatin means that more condensed, like here. Okay, so in the case of the heterochromatin, they stains very darker, like this. Okay, so it means that because heterochromatin is more condensed, it means that there is the inactive transcription. However, euchromatin means that they are less condensed. Okay, if we stain the euchromatin, there is no staining like this. They stains very light. Okay, so in the case of the euchromatin, so there is the active transcription happens. Okay, so it means that epigenetic changes, so when epigenetic changes happens, when they make the heterochromatins, there is an inactive transcription. Okay, when chromatins become relaxed, there is the active transcription is happened. Okay, so in this slide we will learn the fifth subject, eukaryotic gene expression can be regulated after transcription. Okay, so previously we learned this one. Okay, so this process happens, we call it as processing control. This happens after transcription. Okay, so there are lots of processes happens during the processing control. One example is alternative splicing. So what is alternative splicing? The alternative splicing is some exon can also be spliced out, resulting in different mRNA from same gene. Okay, so this is one example. This gene is composed of the exon 1, intron, exon 2, intron, exon 3 is like this. So after transcription, it will make the primary transcriptor composed of exon 1, intron, exon 2, intron, exon 3. After that, alternative splicing happens. So one case, they will make the major mRNA, which is composed of the only exon 1, exon 2, exon 4, exon 5, exon 6. So in this case, this exon is translated, it will make protein 1. Another case is that alternative splicing happens, they will make the exon 1, exon 3, exon 5, exon 6. It can be translated, it will make the different proteins. So final case is the alternative splicing happens. They will have exon 1, exon 3, exon 4, exon 5, and exon 6. So it can be translated, it will make the different proteins. So it means that through alternative splicing, different proteins can be made from the same gene. Okay, so this method is how the eukaryotic gene expression can be regulated. Okay, so alternative splicing happens, it can make the different state, and also different state of mature mRNA will make the different proteins like this. Okay. Okay, so in this slide, I will explain how eukaryotic gene expression can be regulated after transcription. There is another case. So we call it as mRNA inhibition by RNase. Okay, there are two cases. First one is done by microRNA. Another case is done by small interfering RNAs, siRNAs. Okay, let me show you one example, microRNA. Okay, so microRNA, there are several steps there, but today I will not explain it very detailed. The important thing is that microRNA is bind with the RISC complex, so it binds to the mRNA. So it means that this binding, it binds to target mRNA. Okay, so this binding is inhibit translation. So this is belong to the translational control. Okay, and how about small interfering RNA? So this is the small interfering RNA. So I will not explain in details. The important thing is that small interfering RNA is bind to the RISC complex, and this is bind to the target mRNA. Okay, after binding, this binding causes this mRNA degradation. Okay, after siRNA and the RISC complex bind to target mRNA, so it induced the mRNA degradation. So it means that it controlled mRNA stability. Okay, so after transcription, mRNA inhibition can be happens. For example, due to the microRNA or due to small interfering RNA. So main difference is that microRNA inhibit translation. However, the small interfering RNA is they induce mRNA degradation. So this is involved in the mRNA stability control. Okay, both of them happens after transcription, but a little bit different. Okay. So next is that proteasome break down the proteins. Okay, this step is belong to the control of protein degradation. Okay, so usually the ubiquitin, this ubiquitin is a target proteins for degradation. Okay, so in our body we have lots of protein. So when the protein is not functional, ubiquitin knows that. So ubiquitin target proteins for degradation. Okay, after that, this complex, okay, proteasome recognized the targeted proteins. Okay, after that, the proteasome hydrolyzed the targeted proteins. So it will remove the targeted proteins. So this is the eukaryotic gene expression regulation after transcription. So we call it as a control of protein degradation. Okay, so the ubiquitin target the proteins for degradation. Okay, when protein is not functional, ubiquitin knows that. After that, this target gene is recognized by proteasome. After that, proteasome can hydrolyze these target proteins. Okay, so this is the final case of eukaryotic gene expression can be regulated. Okay.